Hello everyone, I hope you're all doing great. You're all safe and sound in your place in this lockdown period. Well, in this last video, if you remember, I promised that I'll be making a next video with C4 plants anatomy. So here it is for you. So again, I have taken this topic from the chapter photosynthesis, right? So before we start with the C4 plants, the anatomy of the C4 plants, you can see this video is being played here in the screen in front of you. This is the video that I've made on photorespiration. I would like to suggest you, those who haven't gone through the video, please go through that video so that things will become more easier for you to understand here what all things I'm going to talk about. Right? All right. So let's start with the C4 plants. So I've written a few points. So we will try to talk about all these points which are given in the Cambridge syllabus. I will try to adhere to all those points as much as possible. First is, what are the C4 plants? Understand one thing. C4 plants means these plants, while fixing the carbon fixation is nothing but photosynthesis, right? So while fixing the carbon, the first stable compound they're going to form will be a 4 carbon compound. That means in one molecule of that organic compound, 4 carbon atom will be present. That's why it is said to be C4 plants. And the kind of cycles, the kind of chemical reactions will be happening in the cyclic process will be known as C4 cycle. Similarly, if you have gone through C3 cycle, you know it better, because in the C3 cycle, the first stable carbon compound which is formed, which is none other than a 3 carbon compound. That's why the name has come up like this. Similarly, in my previous video, we talked about the photorespiration, which is alternatively known as C2 cycle. The reason is same. The first stable compound that's formed in that particular cyclic process is a C2 cycle. It's a C2 carbon compound, right? So if you see those examples, who all fall into this category, understand one thing: near about 1,500 species around us belongs to this category. They are said to be C4 plants. So if I take these examples, you can take maize as an example, you can take sugarcane as an example, then you can take sorghum, you can take millet. These are all different kinds of examples that you can take for C4 plants examples. Now the question is, fine, we have understood who all are there in the C4 plants category, but why we are actually talking about how special the kind of photosynthesis reaction in the C4 plants are, right? Understand one thing. If you remember, we talked about the normal cycle, the normal process that we have seen in C3 cycle, that is the dark reaction, that is a Kelvin cycle. Now in the C3 cycle, if you see, this is a normal transverse section of leaf which are visible in C3 cycle and C3 plants, I'm sorry. And this is a transverse section of the leaf of a plant, which is in case of a C4 plant. Now see, there is a difference in the structure. There is a difference in the adaptability, right? These C4 plants are the plants which actually grow in the tropical and subtropical region where the temperature in most of the cases are high. And these are the plants which are exposed towards a high light intensity, right? So keeping all these factors in mind, they have come up with a specific adaptive feature which is visible here. As you can see, they have come up with a ring like structure here surrounding this vascular element. So this is the vascular bundle inside. So this ring like structure, which is definitely made up of some cells, so it forms like a ring. That's why this kind of adaptation here is said to be Kranz anatomy. Kranz, Kranz anatomy. This Kranz anatomical feature is a characteristic feature for C4 plants because that's where they are needed. C3 plants, they don't need to have this kind of anatomy, so you can't see this kind of Kranz anatomy here. This is one thing. Kranz anatomy is a name. That's it. Now, in the coming slides, we will be talking about that ring structure that I have highlighted just now, where we'll try to see what all cells are there and how come the adaptation comes into play in that particular scenario, right? Now, yes. Now, if you see in this case, this is a typical transverse section of a C4 plant's leaf. Now, see, these are the cells which are present in ring, and these cells are said to be bundle sheath cells. It's special cells which are present surrounding the vascular bundle. So definitely this will be a vascular bundle, right? Surrounding this bundle sheath cells, you have the rings of mesophyll cells. Now, children, not to forget, these mesophyll cells are the photosynthetic cells. That means these are the cells where the photosynthesis can take place. That means these are the cells where Rubisco will be found. Now, like we discussed in the other video, it is the Rubisco who actually plays as a villain, main villain. Same problem happens here because Rubisco shows the dual nature. It can show an affinity towards the carbon dioxide. It can even show an affinity towards oxygen. That's why the name has come up like, ribulose 1,5 biphosphate carboxylase oxygenase. So it can perform carboxylation, it can perform oxygenation under different circumstances. So the kind of circumstances, the kind of environment these kind of plants grow, the C4 plants, they may show, or I should say in most of the cases, they tend to show an affinity towards oxygen. Because in most of the time, they will try to shut their stomata just because they are having, they are living in a heavy temperature region. That's how we define the tropical and subtropical region as. So the problem starts with Rubisco. That means the moment Rubisco sense there is a little high amount of oxygen than carbon dioxide, they will start showing oxygenation rather than carboxylation. As a result of which, those plants will not be in a position to perform efficient C3 cycle. So what will be the loss in the long run? The amount of glucose production will be less. And in the long run, the plant will die eventually. So now if you see this beautiful evolution of the plant which has been taking place due to this condition is this. So what has been done here in this evolution process? From these mesophyll cells, they have taken or they have removed all the Rubisco enzymes. So what's the plus point? No Rubisco, no dual affinity. No dual affinity means no matter if you are having a slight, more amount of oxygen than carbon dioxide, it will not be going to perform the oxygenation because you don't have that responsible enzyme. So no Rubisco. But hey, hold on. I know what exactly you are thinking because it is the same Rubisco who will perform the C3 cycle. Because the first enzyme which plays the most important role in the fixation of the carbon is the Rubisco. So what will happen if you take all the Rubiscos out from the mesophyll cells? But even before that, understand one thing. If you are taking all the Rubiscos out of mesophyll cells, where we are going to place them? We are going to place this Rubisco enzyme inside the bundle sheath cells. So if you see, these bundle sheath cells are actually away from the environment. So where is the environment? Environment will be here. This is where you can see the environment because this is the upper epidermis. So this is definitely the environment, right? And this is the location of the bundle sheath cells. So they are pretty away from the atmosphere. So one good thing is they are not in touch with the atmosphere. This is one thing. Second, the problem still is there. What's the problem? Since you don't have a Rubisco, then who will be there to fix the carbon? Because after all, photosynthesis means fixation of the carbon. So what I can do is, what if I take the carbon dioxide and what if I fix the carbon dioxide at this level, mesophyll level, and what if I send those fixed carbon inside the bundle sheath cells? What do you think? Will these bundle sheath cells ever be run out of carbon dioxide? Never. They will never run. They will never, fall short of this carbon dioxide. But for that, we need to have an enzyme. Now, here comes the second enzyme, which is very important for C4 plants, and the name of the enzymes which are present in the mesophyll cells in place of Rubisco, and they are known as PEP. It has got a name, carboxylase. Now students, see this. Focus on the name here. PEP carboxylase. That means this particular enzyme can perform only one function, that is carboxylation. That means no matter if you are giving them a very least quantity of carbon dioxide, still it will perform carboxylation, right? So that means in the surrounding atmosphere, even if the amount of carbon dioxide is very less, even if the carbon dioxide concentration in the surrounding cells are also very less, this particular enzyme is going to perform only one task, that is a carboxylation. That means they will keep on adding carbon dioxide. And after doing the carboxylation, all those fixed carbon, they are going to send inside the bundle sheath cells. So what is the moral of the story? These bundle sheath cells will never going to fall short of carbon dioxide. Never ever. Are you understanding the point? So what is the plus point of having PEP carboxylase over Rubisco? The Rubisco will show dual nature of affinity. So that means if by chance there is a little bit more amount of oxygen than carbon dioxide, it will start showing affinity towards oxygen than carbon dioxide. So that will lead to the inefficiency as far as photosynthesis is concerned. But here in case of carboxylase, no matter if you're having say 0.1%, 0.2%, 0.3%, very less amount of carbon dioxide, still it will perform only and only carboxylation. You have to take in this way. So all the carbon dioxide will be concentrated at that mesophyll cell with the presence of PEP carboxylase, and those concentrated carbon dioxide that you have, you have gathered the carbon dioxide from different places, you have gathered it in a single place, and you're going to send all those carbon dioxide inside. Now, who is present inside? The bundle sheath cells. These are the bundle sheath cells where you have placed the Rubisco that you have taken out from the mesophyll cells. That means these bundle sheath cells can perform Kelvin cycle. Why they can perform Kelvin cycle? Because they do have Rubisco. What about the mesophyll cells in these particular plants? They cannot carry out Kelvin cycle. Why they can't? Because they don't have the Rubisco. Where are the Rubiscos? You have taken out all the Rubiscos from the mesophyll cells and you have placed it deep inside the bundle sheath cells. Then who will be performing carboxylation there in the mesophyll cells? PEP carboxylase. PEP. The name stands for phosphoenol pyruvate. Right? That's about it. That's about the anatomy of this particular, what you call, the C4 plants. This kind of appearance will give you a ring-like appearance. That's why it is said to be Cran's anatomy. I hope that makes sense. One more thing you have to remember: these bundle sheath cells, they don't perform any light-dependent process. So, what is the moral of the story? That means these bundle sheath cells cannot produce oxygen. Understand one thing, because it is a light dependent phase where you are going to produce oxygen, right? The photolysis. But here in the bundle sheath cells, it doesn't have any grana. Their chloroplasts are agranal, so they cannot perform light dependent reactions, right? But if you come here in the mesophyll cells, these mesophyll cells are having chloroplast, which will definitely have grana, so they can perform the light dependent phase. So once again, please understand one thing: mesophyll cells, they cannot perform Kelvin cycle, but they can perform light dependent phase. Bundle sheath cells, they can perform Kelvin cycle, but they cannot perform light dependent phase. Right? The enzymes which are taking place, which are replacing the Rubisco at the level of mesophyll cells, are the PEP carboxylase. So whatever Rubisco was there, you have taken out all the Rubisco and you have kept them back inside the bundle sheath cells. Right? That's it, guys. In the next video, we will be talking about this particular C4 cycle very quickly. So that's all for the day. Right? I hope you have understood and enjoyed the video. So if you have any doubt, any kind of doubt that regarding what you call this anatomy of the C4 plants, please do not forget to write in the description below. And Please, you can connect with me in the possible platform which I have mentioned here, as you can see, the Facebook, Twitter, LinkedIn, and Instagram. So all the links for this particular social networking sites are given in the description below. So I hope you have enjoyed the video and stay safe, be at home. I know it's a tough time for all of us, but we will overcome definitely. We have to overcome. You have to be positive mentally, right? Anyways, that's all for the day. In the next video, which will be coming very soon, we will be talking about the C4 cycle, right? That's all for the day. Take care. Bye bye.